I love working out. It's one of my favorite things to do. But have you ever wondered how our muscles are able to contract in our everyday lives? Let me introduce you to the sliding filament theory. But before we get into the theory, let's talk about skeletal muscles.
Skeletal muscles are muscles that are attached to our bones and held together by tendons. These muscles produce all the movements in our body, and unlike smooth and cardiac muscles, they're voluntarily controlled. This is a microscopic view of a skeletal muscle. A skeletal muscle is composed of many cells that form a muscle fascicle. A muscle fascicle itself is composed of many muscle fibers. A muscle fiber is composed of long contractile units called myofibril. And finally, myofibrils contain small repeated structures called sarcomere, and it is here where our sliding filament theory will take place. Within the sarcomere, we have two different filaments. Actin are the thin filaments, myosin are the thick filaments. The actin filaments are covered in two proteins, troponin and tropomyosin. These proteins act as a lock. And if we closely look at the myosin or thick filaments, we can see that it's covered in tiny structures named myosin heads. These structures are also important. At the edge of the sarcomere, we have structures called Z-lines. At the center, we have a structure called the M-line. Muscle contraction occurs when the myosin heads and the thick filaments slide and pull the thin filaments closer to the M-line. This is known as the sliding filament theory.
How does this happen?
First, it all starts in the somatic nervous system, with a signal from your brain to your body to move a muscle. This signal comes as a form of an action potential, and this is sent to the motor neurons of your muscle fiber. The place where the axon terminal of the motor neuron and the muscle fiber connect is called the neuromuscular junction, and it's here where a neurotransmitter called acetylcholine will cross and release calcium ions. These calcium ions can then get sent to the sarcomere to unlock and deactivate the troponin and tropomyosin. The actin site is now unlocked. Even though the actin site is unlocked, the myosin heads can't bind just yet because we need a secret sauce.
That secret sauce, ladies and gentlemen, is ATP.
As the ATP molecule attaches to the myosin head, it hydrolyzes and becomes ADP plus phosphate. Myosin head can now bind to the actin filament in a process called a cross bridge and perform a power stroke. As long as there's adequate levels of calcium and ATP stored, this process can be repeated again, and that's the sliding filament theory.